Hello everyone. Thank you for joining us today. It's Scholar Avenue. Today we are going to talk about gene regulation in eukaryotes. Gene regulation in eukaryotes refers to the mechanisms by which gene expression is controlled or modulated. In these organisms, eukaryotic gene regulation is a complex process that involves various levels of control, allowing cells to respond to their environment and develop into different cell types with distinct functions. Here are some key aspects of gene regulation in eukaryotes. First is DNA structure and chromatin remodeling. The first level of gene regulation occurs at the level of DNA. The DNA is wrapped around proteins called histones to form a structure called chromatin. The accessibility of genes within the chromatin can be modulated by various processes collectively known as chromatin remodeling. These processes involve modifications to the histones, such as acetylation, methylation, and phosphorylation, which can either promote or inhibit gene expression. Second is transcriptional regulation. The primary level of gene regulation in eukaryotes is at the transcriptional level. Transcription is the process by which the DNA sequence of a gene is used as a template to produce an RNA molecule. Transcriptional regulation involves the binding of specific proteins called transcription factors to regulatory regions of the DNA, such as promoters and enhancers. Transcription factors can either activate or repress gene expression by facilitating or blocking the binding of RNA polymerase, the enzyme responsible for synthesizing RNA. Third is RNA processing. Eukaryotic genes often undergo several processing steps after transcription to produce a mature RNA molecule. These steps include 5-capping, splicing, and 3-polyadenylation. Regulation can occur at these processing steps, allowing for the production of different RNA isoforms from a single gene or controlling the stability and translation efficiency of the RNA. Number 4 is RNA stability and degradation. The stability of RNA molecules can be regulated by specific RNA-binding proteins and non-coding RNAs. These factors can influence the half-life of the RNA, determining how long it persists in the cell and whether it is translated into a protein. Fifth is the post-transcriptional regulation. Once an RNA molecule is produced, additional regulatory mechanisms can control its translation into a protein. These mechanisms include the binding of regulatory proteins to the mRNA, as well as the action of small non-coding RNAs, such as microRNAs and small interfering RNAs. These molecules can inhibit translation or promote the degradation of specific mRNAs. Sixth is epigenetic regulation. Epigenetic modifications refer to heritable changes in gene expression that do not involve changes to the underlying DNA sequence. These modifications, including DNA methylation and histone modifications, can be inherited through cell divisions and can have long-lasting effects on gene expression patterns. And the last one is cell-specific and developmental regulation. Gene regulation in eukaryotes is highly dynamic and can vary between different cell types and during development. Different combinations of transcription factors and epigenetic modifications contribute to the establishment and maintenance of cell-specific gene expression programs. Overall, gene regulation in eukaryotes is a complex and tightly regulated process that involves multiple levels of control to ensure precise and coordinated gene expression patterns in different cells and tissues. Thank you for watching, and don't forget to subscribe, like, and share.